Hello everyone. So today I'm going to be guiding you through the Visual QC functionality that we've got built directly into our SpotMap MS software. Now, QC and QA, the quality of the data that you're getting out of our software is paramount to us here at Total Lab, and that's why we've built a lot of features around that. Visual QC is just one of them. And it's built into the software because LC-MS data quality has long been an Achilles heel of the process, in that you get a lot of data out of these machines and it can be very difficult to tell what is good quality data versus what is bad quality data until you've had to go through the whole process of analyzing it and you realize at the end, oh, something must be wrong because this doesn't make sense, and then work back and realize actually from the beginning you've had poor quality data. Wastes a lot of operator time, wastes a lot of farmer time. So the QC features that we've built into our software are designed to avoid that, or at least alert the users as soon as possible with a visual view that the data, what the data looks like, for them to make the judgment, say, actually, I don't trust this data, I'm not confident in this data quality, I'm not going to waste the time analyzing it. And in fact, I've still got my sample, defrosted on my desk. I'm going to go and run it again and kind of cut out all that wasted time from the classical method of getting to the end and only observing the error then. So I'll show you the QC, I'll kind of guide you through it, and I'll explain to you why we show it as soon as humanly possible. So I'm just going to open an existing experiment that we've got here, a little demo experiment that we've got built directly into the software. So here the software is processing, it's loading the raw trace files in, and as you can see, the first thing we show users whilst it's still loading, so this is still processing, it's still processing the data, but as soon as we can, we show users that we've got this visual QC going on. So this is what the visual QC section of the software looks like. You can see that we've got our mass charge ratio on the left-hand side, on the left axis, and our retention time here in minutes on the x-axis. I may have got those the wrong way around, I can't quite remember. So you can see here, this is a visual view of basically mass overcharge versus retention time, and these should look, for this experiment, these are replicates. These should look similar, and if you're running a very well-characterized workflow, shall we say, these should look very similar every time. So if you're running the same sample day in, day out, these patterns, you should be able to recognize them. But it's also within these that you would be able to spot any immediate errors in your sample, in the way that the machine has kind of dealt with your sample. And that'll make sense in a second when I show you a classic example of it going wrong and what the outcomes of that were. So as you can see here, all of these samples pretty much line up with each other. And again, it's the same sample run 3 times, so that's what you would expect. So this is the first QC metric that users have got to go off within the software to make the decision as to whether they want to analyze this data set or not. From the top here, we've got some different QC measures as well. I might make a separate video on those, in fact. On the bottom here, we've got a contrast range, so we can change the contrast of these particular areas. So the darkness of each area is relative to the intensity in that space, in that kind of XY coordinate. But you can change the contrast and maybe highlight some areas so you can see it a lot. There's a lot of focus around this retention time at this mass overcharge point. If we bring the contrast right down, you can see it's much higher than the rest of the trace. But again, this graph is not designed to show you the minutiae of kind of everything that you've gone through. It's to give you a very quick visual check to say these things look like I expected and they look like each other. Now I'm going to show you how the visual QC can be used in an instance when an experiment has gone wrong. So you can see this is a different experiment that we actually commissioned as part of the development of SpotMap MS. And these again are the same sample repeated 3 times, but you can see within the visual QC section, we've got some similarities, but some significant differences in areas of the visual QC. Now we can see these are largely similar, although we've got these white lines, this banding going along. But certainly for sample number 3, that has eluted much quicker than the other 2 traces that we're looking at here. Now, this indicates to us that perhaps this run has not gone correctly. Something's gone wrong and we need to repeat it. Now, when we were speaking to the people that performed this work for us, in fact, and showed them this, they in fact said with the software that they were currently using, they never would have seen this. So what they would have done is go through the whole experiment, analyze it, get to the end, get their data processed and say, why has one of our replicates, why are the results so different for this replicate? However, using SpotMap MS, they're now able to see that information straight away and actually say, oh, we should run the sample again and discount this trace and get ahead of that and not waste all of that time processing it and trying to work back from an erroneous result. They can see that straight away, so it saves them a huge amount of time, or it would have saved them a huge amount of time if they were using SpotMap MS. The hope is that it's going to save you guys, the pharmaceutical industry, a huge amount of time as well within, LC-MS based host cell protein analysis, along with all of the other QC parameters and automation functionality within the SpotMap MS software. Now, if you're interested in talking to us about SpotMap MS, getting a free trial of SpotMap MS to see how it works on your data, or learning more about the product or learning more about Total Lab as a company, please check out the links in the description below.